Good morning all. Today we are going to discuss about mechanism of ATP synthesis. So we have already gone through different steps in glycolysis as well as TCA cycle, and also we have studied about the electron transport chain, etc. So during the various steps in glycolysis and the citric acid cycle, the oxidation of certain intermediate precursor molecules occurs, and it may cause what? The reduction of NAD plus to NADH to H plus, NADH plus H plus, and also another intermediate, that is FAD to FADH2. NAD is nothing but nicotinamide adenine dinucleotide. FAD means flavin adenine dinucleotide. So this both NADH and FADH2, they transfer protons and electrons to what? Electron transport chain. And this may produce what? Additional ATPs. And by transferring these protons, the ATP molecules are produced by oxidative phosphorylation that we have already studied. So here we are going to discuss now what is the principle or hypothesis behind the ATP synthesis. During the process of aerobic respiration, coupled oxidation reduction reactions occur, and electron carriers are often part of what is called an electron transport chain, which is a series of electron carriers that carry these electrons which are transferred by the intermediates, and finally they transfer those electrons to oxygen, which is the terminal electron acceptor, and finally ATP molecule is synthesized, that we have already discussed. So this ATP, that is adenosine triphosphate synthesis. So this, for the ATP synthesis, they require an enzyme system which is called ATP synthesis. And they are nothing but it is a multi-subunit protein complex. Multi-subunit protein complex that use an electrochemical proton motive force across the membrane to make the cell supply of ATP. So it uses the electrochemical proton motive force across the membrane, and which helps to supply the cell what? ATP from adenosine diphosphate, that is from ADP and inorganic phosphate PI. So this enzyme, which one ATP synthesis, are found in bacteria as well as in chloroplast as monomers. But in mitochondria, they can be seen as a row of dimers. Actually, this mitochondria is present in eukaryotes, and that bend the inner membrane to facilitate the mitochondrial cristae. Next is, we can discuss another term called chemiosmosis. And this chemiosmosis, it is the term which are involved in the ATP formation. It is the movement of ions across a semi-permeable membrane-bound structure down their electrochemical gradient. An example of this would be the formation of ATP by the movement of hydrogen ions across the membrane during the cellular respiration or photosynthesis. So the movement of the hydrogen ions across the membrane during either cellular respiration such as TCA cycle or photosynthesis. And that mechanism is called chemiosmosis, movement of ions across the semi-permeable membrane. Hydrogen ions or protons will diffuse from an area of high proton concentration. So how will be the movement occurs? It can be moved from the area of higher proton concentration to an area of lower proton concentration. So during their movement, an electrochemical concentration gradient can be established. The electrochemical concentration gradient of protons across the membrane can be harnessed to make ATP. And the process is related to what? osmosis, the diffusion of the water across the membrane. So because of this similarity with the osmosis, it is called chemiosmosis. So the ATP synthase is the key enzyme that makes ATP by this chemiosmosis. And this allows the protons to pass through the membrane and uses the free energy difference. Free energy difference to phosphorylate what? ADP, and makes ADP to convert ADP to ATP. The generation of ATP molecule by the chemiosmosis occurs in mitochondria and in chloroplast. They may also occur in bacteria and archaea. And an electron transport chain pumps H plus ions in the thylakoid spaces through the thylakoid membrane to stroma, and this occurs in eukaryotes. The energy from the electron movement through electron transport chain crosses through the ATP synthase, and which allows the proton to pass through them and use this free energy difference. What? This free energy difference is used to phosphorylate what? ADP, and thus the A can be converted to ATP. So the energy from the electron movement through electron transport chain goes through the ATP synthase. Next is about chemiosmotic hypothesis. The chemiosmotic hypothesis, it was first proposed by Peter Mitchell. The hypothesis states that a proton motive force was responsible for driving ATP synthesis. And in this hypothesis, protons would be pumped across the inner mitochondrial membrane as electrons went through the electron transfer chain. So in this hypothesis, what happens? Protons would be pumped across the inner mitochondrial membrane. And this may result in a proton gradient can be established with a lower pH in the intermembrane space and an elevated pH in the matrix of mitochondria. So a proton gradient can be established within the intermembrane space and the matrix of the mitochondria. And in intact inner mitochondrial membrane, actually it is impermeable to protons, and it is a requirement of such a model. A proton gradient and membrane potential both are what? Proton motive force. And this proton motive force are used to drive ATP synthesis. And this process, such as mitochondrial membrane, this may occur in eukaryotes. In effect, the pH gradient acts as a battery which stores energy to produce ATP, this pH gradient. Over the past several years, Mitchell's chemiosmotic hypothesis has been widely accepted as the mechanism of coupling of electron transport and ATP synthesis. He was awarded Nobel Prize in Chemistry in 1978. This acceptance by the scientific community is a result of accumulating experimental evidence supporting the hypothesis. Now, what are some of the evidence supporting the Mitchell's chemiosmotic hypothesis? Electron transport generates a proton gradient that we already know, and the pH measured on the outside, it is lower than that measured inside. The pH is lower on the outside than the inside. Only the proton gradient established, it is needed to synthesize what? ATP. The electron transport is not required as long as there is another mechanism for generating pH gradient. So, a reconstitution experiment carried out by Racker and Stokenius, they also catalyzed light-driven proton uptake of. They showed that the generation of a proton gradient can result in ATP synthesis in a totally artificial system. So they both tested in the artificial system as well as natural. So they both found that this proton gradient is necessary for the ATP synthesis. So these are the experiment, that is a mitochondrial ATPase complex from the beef heart inserted to an artificial lipid bilayer, and inserted this bilayer was a membrane fragment containing protein, bacteriorhodopsin. And these are all the experiment conducted, and the electron transfer chain and ATPases are asymmetrical oriented in the inner mitochondrial membrane. So in this experiment, it was understood that a net gradient of protons are essential, which is called proton motive forces that are required for the synthesis of ATP. Compounds called uncouplers were found to collapse pH gradient. So there are certain compounds called uncouplers which may collapse the establishing the pH gradient by shuttling the protons back across the membrane through the compounds. And one such uncoupler is dinitrophenol. In the presence of this uncoupler, what happens? Electron transport chain continues, but no ATP synthesis occurs. That's the difference. This is the dinitrophenol molecule. So what happens in prokaryotes? How this chemiosmotic mechanism of ATP synthesis occurs? The bacteria and archaea can use this chemiosmosis to generate ATP that we have already discussed. Cyanobacteria, green sulfur bacteria and purple bacteria, these are all coming under bacteria. They synthesize ATP by a process called photophosphorylation. So this green sulfur bacteria, etc., they utilize light, that is in the form of photosynthesis. This bacteria uses the energy of light. So this type of bacteria uses light energy to create a proton gradient using a photosynthetic electron transport chain. Non-photosynthetic bacteria are also present, such as E. coli. This E. coli also possesses what? ATP synthase. In fact, the mitochondria and chloroplasts are also the product of endosymbiosis, and they trace back to incorporated prokaryotes. So this process can be described as endosymbiotic theory. The origin of mitochondrion triggered the origin of eukaryotes, and the origin of plastid, the origin of archaeplastida, one of the major eukaryotic subgroups. So the chemiosmotic phosphorylation, it is the third pathway that produces ATP from inorganic phosphate and an ADP molecule. This process is the part of oxidative phosphorylation. So this is the picture of this oxidative chemiosmotic coupling between the energy of sunlight. This is the sunlight, and this is the bacteriorhodopsin pigment. This is the ATP synthase. Cytoplasm, ADP plus Pi forms ATP, and this cytoplasm. These are the H plus ions. So this ATP synthase release H plus ions, etc. So these are seen in the certain bacteria such as photosynthetic bacteria. So these are all about chemiosmotic mechanism of ATP synthesis. Thank you.